Good day. I'm Martin Gagel with Market Radius Research. It's Wednesday, February the 9th, and we've got CEO Frank Gleeson of Satellos Bioscience. Satellos is a drug developer targeting Duchenne muscular dystrophy by leveraging new research on how muscle stem cells regulate muscle repair and growth. I've got Satellos on the call for several reasons. I've been a shareholder of ICO Therapeutics, through which Satellos and its team went public in August of last year, raising over $7 million and restructuring the company. So I'm a shareholder. I recently met Frank and was very impressed by his team and the company's qualifications and experience. I'm a sucker for investing in companies with innovative and new technology that are making big swings to make a big impact. With this meeting, I'm hoping to better understand the science and the drug development pathway, milestones, and inflection point expected over the coming months and years. But remember, this is neither a recommendation nor investment advice. We're here to learn about the company, do your due diligence, and come to your own investment conclusions. Frank, thank you very much for joining us today. And please tell us what's going on at Satellos.
Well, thanks, Martin. Thank you for the introduction and thank you for the invitation. I'm delighted to be here today and to speak with your network about Satellos and let them know what we're doing and where we're going with the company. Our business is about rebuilding muscle from within. So we created that tagline because it aptly describes what we're trying to do. Our goal is to develop new medicines that can be taken by patients who suffer from serious muscle disorders to enable their bodies to reset their own ability to repair and grow muscle. And so I'll tell you about that as we go along. We think this is absolutely fascinating, next generation science, and we're just thrilled to be here. So we are a public company, of course, so be mindful. I will be making forward-looking statements. So one thing that's not a forward-looking statement is that muscle is a regenerative organ, and it has the capacity throughout our lives to continue to regenerate. In fact, without that capacity, we would not be able to grow in size and stature. We would not be able to go to the gym and get bigger. We would not be able to recover from injury. So it's a critical organ in our body, and we are leaders in the world in the understanding of the regenerative process in muscle. Our team, our co-founding scientist, identified muscle stem cells in the body and how they give rise to regeneration. We've discovered dysfunctions that occur in these processes that cause degenerative process, and we've identified corrective mechanisms which the scientists refer to as pathways, biochemical pathways in the body that we can modulate with drugs to correct these deficiencies. Muscle is also the, yes.
So, and just like when you're talking about muscle is a regenerative organ or whatever, that's key to it. Like when you go to the gym and you work out, you lift heavy weights, you actually do a little bit of damage to the muscles, and then the body rebuilds those muscles and actually makes them a little bit stronger through the rebuilding process. Is that sort of a layman's how it works?
That's it exactly. That's exactly what we have. We're deliberately attempting to create micro tears in our muscle, and then that triggers a regenerative process. So even when we do simple things, stand, sit, ride a bicycle, whatever it is, walk upstairs, we're actually always damaging our muscle tissue. And it sends sort of signals, biochemical signals, to stem cells, and I'll show you about stem cells in a moment, to jump into this regeneration process and to create new muscle fiber. And that's exactly how you get bigger at the gym. In fact, anyone who goes to the gym for that purpose knows you don't work the same muscles 2-3 days in a row. You rotate around and you have a different routine for arms and legs one day and back and chest another day and so on, because you need to give your body time to go into the repair and regeneration process. And if you don't, you exhaust that capacity. And in some diseases, genetic diseases, all the way through to aging, comorbidities associated with cancer, this process either fails or gets exhausted, or in other ways is impaired. And unfortunately, these diseases, which are all too common, and we can all think of people, I'm sure, who have had trouble recovering from surgery or have been in the ICU for other reasons, and their muscles atrophy. These musculoskeletal diseases are not well treated. They're not well served by current medicines. And the market is massive. So we're talking a $20 billion market by a number of different estimates, growing at double-digit rates. So big opportunity, not well served, and we have a very unique understanding of the processes at work to develop medicine. Now, my co-founding partner, I also founded the company in 2018 along with Dr. Michael Rudnicki, who's based in Ottawa. Michael and I have known each other 20 years. This is the second company we've done together. He's an Order of Canada, member of the Royal Society of London, England, chair of the Stem Cell Network of Canada, and scientific director. That's how we first met, through that body which funds stem cell research in Canada. He has consistently and regularly published throughout his career in the highest of scientific journals: Cell, Cell Stem Cell, Nature. These are the elite of the elite. And from his first publications back around the year 2000, when he established his own lab in Ottawa, he has been at the forefront of identifying muscle stem cells, and those particular cells within a special niche in the muscle that give rise to regeneration, and also determining how that regeneration goes on, and then linking and proving that muscle loss in aging is caused by breakdowns in how muscle stem cells function, and that more recently in the disease Duchenne muscular dystrophy, which is a genetic disorder I'll tell you more about, that muscle stem cells actually cause the progressive loss of muscle that these boys experience because the stem cells aren't working properly. So how that all functions is described on this slide. So if you look at the image on the upper left, the muscle stem cell image, that little black micrographic picture is of a stem cell on a muscle fiber dividing. The writing underneath is for the scientific junkies. It characterizes how that particular stem cell is recognized by microbiologists that study, molecular biologists that study it. And the stem cell itself lives deep within the muscle, and it lives next to the fiber under the basal lamina. And its purpose in life is only to do one thing, and that's to divide at the right time in response to cues from its environment. And when it divides, which is the image below, what it does is it divides to create two types of cells. It's in its own type of cell, a muscle stem cell, for self-renewal to keep the pool of stem cells going, and a differentiated cell called a muscle progenitor cell. So this is the fascinating thing about a stem cell, is that it has to create one copy of self and one non-self. And it's that differentiated cell that goes on to become muscle. And in diseases or disorders of aging, frailty, some comorbidities, muscle loss due to surgery and so on, we lose stem cells. So we don't have enough stem cells for self-renewal to keep the regeneration cycle going. And that's why as we get older, our muscles become more frail and they become more difficult to regenerate. And we lose muscle mass, as you undoubtedly know from parents and grandparents. And in the case of Duchenne muscular dystrophy, a different phenomena happens that Michael discovered and which shocked the scientific world when it was published 5 years ago, or 6 years ago. That's that in Duchenne muscular dystrophy, which was thought to be solely a disease of the muscle fiber, what Michael discovered is that the body is no longer producing enough muscle progenitor cells to repair the damage to the muscles of these children. And so what happens is you get an accumulation, or what appears to be an accumulation, of too many muscle stem cells, the purple ones, and not enough differentiated muscle progenitor cells, the green ones. And that phenomenon is at heart of many, many disorders related to dystrophies, related to atrophy, and we are now building the capability to treat those. So
Frank, could you go back one slide? I just want to ask and clarify a couple things.
Yeah.
So with the stem cells, it splits or it replicates and it creates a progenitor and one of itself, so it can keep doing that. But in Duchenne muscular dystrophy, it just creates another copy of itself and doesn't create a progenitor cell, which is the one that actually builds the muscle or becomes the muscle?
Yeah, so what's happening at the level of the molecular biology under the microscope, if you looked at it, is that the stem cell is attempting to divide to create a self and a differentiated cell, a progenitor cell. It's attempting to do that. And when it does that, it physically changes its position on the muscle fiber. It's called its polarity. It reorients itself so that it can create two cells that are different. It's a really elegant and fascinating process. And in the process of doing that, because of this particular disease and what's happened genetically, those cells are unable to figure out up from down. And so they're attempting to divide and create two cells, and the wiring gets screwed up, and basically they just keep repeating themselves. That's essentially what happens. They're attempting and they're failing in the attempt. And
is this a genetic cause, or is there some sort of external factor or some other factor?
Well, ultimately, if you let me go on in another slide, I'll tell you about that. But yes, it is because of a genetic starting point. But the unknown aspect was that starting point affects the stem cells, and that the stem cells can no longer properly divide. And it's almost a seminar of itself on how that happens, but it's really quite fascinating step-by-step science. And what we've discovered is how to overcome that deficit. So we've now identified how to correct that ability and enable these cells to properly orient themselves in order to divide.
One other quick question. You refer to Duchenne muscular dystrophy. As a layman, I've only heard of muscular dystrophy, and I did a little Googling, and there seem to be two types of muscular dystrophy, Duchenne and Becker. Am I right there? And
Yes.
Can you give like 20 seconds on the difference between the two?
So, yes, of course. So these particular disorders are named after the individual that first identified them, the physician in time that first identified them, and then they became eponymous, associated with that physician's name. So Duchenne is the most severe and the most common of all the dystrophies. It essentially is caused by a mutation in the dystrophin gene that results in an absence of the protein. Genes make proteins. So the dystrophin protein is essentially absent in Duchenne patients. Becker is a milder mutation. So the Becker phenomenon is one in which there are mutations to the gene. However, they're not as severe. And so those individuals are able to continue to make a portion, at least, of the protein. So they are on a continuum. If you think of it that way, it's a continuum, with the Duchenne patient being the most severe and the most hard hit. There are a range of other dystrophies. There's limb-girdle muscular dystrophy. There's a facial muscular dystrophy, FSHD. There's a laminin and a collagen type of deficiency that causes dystrophies. So there's a wide range. Myotonic dystrophy is another one, a wide range of dystrophies, all with slightly different characteristics. But we believe virtually all of them at heart have a stem cell problem.
And dystrophy, just to get, so we know what the terms are, that just means like a wasting disease. So muscular dystrophy is muscles wasting. Something else dystrophy is the other thing wasting, right?
Yes,
Okay.
and dystrophy refers, in the case of Duchenne, it refers to the gene. The gene is called dystrophin. And so if you don't have the gene or a defective gene, you have a dystrophy. And then I think that term has become more generically applied.
Thank you.
Okay. And so from a business point of view, I know the science is super fascinating, but there's also a business that we're trying to build, and that's important for us as business people and for you as investors as well, or potential investors. So the focus of the company is going to be initially on rare diseases. So we have built a great body of information and evidence about our potential ability to help Duchenne patients and other dystrophic patients. The need's massive, as I'll tell you about in a moment. And these disorders also, because of their nature and because of the demand from patient groups for treatments, tend to be met with a great deal of receptivity by the FDA and other regulators, which can lead to accelerated pathways to approval and accelerated pathways to getting to market and generating revenues, or leading to M&A activity if that's turns out to be our exit strategy. And then we will broaden as time goes on into broader mass market indications. So there are many diseases associated with aging, frailty, sarcopenia, lots of issues associated with chronic illness, cancer, COPD, and there's increasing very strong evidence that the same type of phenomena we see in some of the dystrophies occur in those illnesses with muscle loss. In fact, in the case of Steve Jobs, it was really muscle loss associated with this pancreatic cancer that killed him before the cancer did. And then other types of muscle loss associated with trauma or surgery. And then we'll establish the conditions for major growth. What we mean by that is that as we move along, we will need to be able to access additional capital markets and so on to do longer term or bigger clinical trials. And so we'll pursue moving to the NASDAQ in due course and expanding our R&D capabilities and physical infrastructure as we grow. And so I'd like to just now move on to telling you about Duchenne muscular dystrophy. We've talked about it a little bit already, so some of the aspects are probably becoming clear to you, but just to reinforce a couple of points. This is a disease in which muscles degenerate and get destroyed progressively through the life of a young person. It affects males. It occurs 1 in 3,500 births worldwide, regardless of geography or race. And it's caused by a mutation in the dystrophin gene, which essentially prevents the body from making the dystrophin protein. And it presents in young boys, it's X-linked, it's carried on the X chromosome, which means that girls have 2 X chromosomes, so they have a backup. Boys don't. So if they get this on their X chromosome, they have the disease. And
Hey, Frank, sorry, does it occur in females as well, but just at a much lower rate, or is it just exclusively males?
Yeah, it's almost non-existent in females. One supposes it could be possible that both alleles of the gene would be defective in a female, but as you'll see, because of the onset, it really renders children almost incapable of any activity beyond their late preteen years. So it's almost inconceivable that it would happen. There are some really genetic freaky situations where there are reported incidents of females with muscular dystrophy, but it's almost an impossibility. But of course, it's never an impossibility in human biology. So the young child will present at about age 2 or 3 with difficulties with mobility, apparent difficulties that the parents then can't miss, so to speak, and take their children to physicians where they're diagnosed in that age group of 2 to 5 with having muscular dystrophy. And they generally wind up losing the ability to walk between the ages of 10 and 12, depending on how aggressively they're put on corticosteroid treatments such as prednisone or derivatives of prednisone. And they wind up in a wheelchair from that point. And the progressive muscle damage really begins to take hold from then on. And ultimately, the disease generally leads to death in the late 20s or in the 30s. And corticosteroid treatment is standard of care to keep them alive as long as possible. And the current treatments that are in the field, and you can Google and you'll find there are many different approaches. And often people say to me, well, that means there's a very crowded field. Well, the thing is that none of these current treatments address regeneration. None of them is focused on the idea of creating new muscle fibers. Every single approach is focused on existing fibers. The very fibers that you will note are the ones being destroyed by the disease. So whether it's gene correction, trying to put the gene back in the fiber, muscle inflammation, trying with corticosteroids to reduce the inflammatory damage, or the muscle fibrosis that infiltrates, or trying to protect the muscle, or get mitochondrial energy sort of redirected to the muscle. All of these approaches treat the existing, declining muscle fiber. We are the only group that's focused on addressing regeneration. And we think that has two very big implications for us. Number one, we're very distinctive, and we have the potential to create a disease-modifying approach that will benefit patients. And two, our approach would be complementary to any of these other approaches. We're not crowding them out. If there's a breakthrough in treating fibrosis and breaking up fibrotic tissue, that would be a benefit to us and to those children, and we could make that success an even bigger success by allowing new muscle to be created. So we are trying to shift the focus away from only treatments that look at damaged fibers to creating new fibers, which of course in the fullness of time would lead to less damaged fibers.
Frank, sorry,
Yeah.
just simplified terms as the existing therapeutics are just trying to slow down the degenerative process, but you're actually trying to increase or have a regenerative process. So theoretically, in the very best case scenario, you could actually kind of cure it. If you're wasting away and your body learns how to build new muscle, you could literally get better.
Yes. I've spent a lot of time in biotech in my life, Martin, and spent a lot of time during the 90s as a venture capitalist investing in cancer companies that all thought they were going to cure something. So I'm reluctant to use the word cure ever, because I think it's from a drug developer point of view, I feel it's not our place to say that. But the clear implication of our work is to make a dramatic improvement in the capability of patients to walk and function normally. And if you have cancer and you stay cancer free for 5 years, they say that's a cure. So if other people want to say that, that's their prerogative. I'm not criticizing you, by the way, for saying that. I'm only saying from my point of view, I don't like to use that word. I like to say that we are going to make a dramatic difference in the functionality of these muscles for these patients. That's our goal.
So in layman, to rephrase in layman's terms, you're hoping to make an improvement in the muscles, not just slow down the degeneration.
Totally. Our goal is to create new muscle. That's the objective. So that the degenerative process doesn't take over. So that's what's happening in these children. Their body is not capable of keeping up with the need. So as they go from being 2-year-olds to 5-year-olds, their body mass increases dramatically. Well, they're not generating enough fibers to keep up with just their growth. Then as they get older and they get more active and they're trying to be boys or kids or do whatever they do, the damage that occurs just in normal living isn't being repaired. So you get this double whammy that they're not keeping up with the growth of their skeleton, and they're not keeping up with the demands placed on their bodies. So they wind up in a situation where they lose mobility, and then gradually they lose ultimately virtually every other function, and generally they die of heart failure.
So Frank, sorry, so if they actually do exercise in that ends up speeding up the degenerative process. So doing less actually is better for them, or it slows it down?
Well, I don't think that's what's recommended, but ultimately they become less mobile, so they are unable to function, and that's just the horror of the disease.
Yeah.
So essentially, what's happening is that what we're trying to depict in this image is that as the demand is placed on the muscle, the stem cell divisions don't work properly. So they're not creating enough new progenitor cells, and regeneration fails. And what we're trying to do is, with a drug, is to correct those divisions, to correct the stem cell divisions so that the body can make progenitor cells and regeneration occurs. And so if regeneration is occurring, then these children can hopefully they can keep up with the demands that their body is placing on them. And the whole concept is depicted here in this experimental work that we did, and a proof of concept that this could work. So the way that this disease is studied at the laboratory level is in a mouse model called the MDX mouse. So this is a mouse that has been genetically altered to have a defect in the dystrophin gene and have the same condition that humans have of muscular dystrophy. And in these mice, they do not produce enough progenitor cells. So that can be cataloged. You can look at that under the microscope. You can count all the cells. So this is factual information. The MDX mouse does not have enough progenitor cells. And so these mice then have difficulty. And the question we asked is, if we treated these animals with drug, could we restore those divisions? In other words, reset this balance so that there is a sufficient number of progenitor cells. And so we treated mice. We treated them for 4 weeks with drug to see what would happen. And the data gets graphed, and you can see very clearly in the graphs, which I'll explain, that the untreated animals, which are the 2 bars on the left, the purple bar represents the muscle stem cells, and the green bar represents the progenitors. So you can see in the untreated animals the same imbalance that's depicted in the picture above, in the cartoon picture above. After treatment with drug, we shift that balance. So now the treated animals are producing a sufficient number of progenitor cells to repair themselves. And then we look at the muscle. So when you look at the muscle, in this case, we looked at their diaphragms, which might be the single most important muscle to look at in a lot of ways because of its importance in respiration. When you think that at the end of the day, these children ultimately succumb to heart failure. So we see bigger muscles. So you're looking at these muscles, and you can see the overall diameter of the muscle on the right is greater than the muscle on the left, the treated versus the untreated. So it's a bigger muscle. That's good. You have less fibrosis. So the green measures fibrosis. It's a stain that picks up the fibrotic tissue. And you can see that the fibers, the individual fibers that make up the muscle, which are the black holes, so does the cavities, the black cavities, they're not actually cavities, that's the muscle fiber. So think of this as a reverse out image, are actually larger. So The next question we ask then is, of course, does it lead to better performance? So what we see is that in these graphs on the left, you're looking again at untreated versus treated. The animals run further, they run faster, they run for more time, and they're stronger, which is the graphs on the right. So this provides proof of concept in a gold standard model of Duchenne that our thesis that A, we can correct this deficit of progenitor cells. Number one, we can do that. And two, that leads to a higher quality of muscle and enhanced performance. Now, what I want to show you on the next slide is some information that has just been published over the weekend, last weekend, that had us massively excited. This is work that was done by a different group than us. This is work that I'm going to show you that was done in the United States at the Ohio State University Nationwide Children's Hospital. This is probably the largest center in the world for the genetic study of Duchenne muscular dystrophy. And this is a group of geneticists who did this study. And I can tell you, without any lie at all, that these people are pure geneticists who don't really believe in our theory. They believe in genetics as the answer. So I only say that because once you see what they've concluded, you may understand a little more why we're so excited. So I'd like to draw your attention to the graph on the left. What this group of geneticists did was they looked at a group of 419 boys, many of whom were able to walk well past their peer group. You recall that I mentioned that generally between the ages of 10 and 12, that's when the vast majority of Duchenne children are in a wheelchair. Well, what this group identified was a proportion of these boys that were walking well past ages 10 to 12, and we've highlighted those with the blue box. Some of them almost to the age of 30. And there was no obvious explanation for this. These boys did not have dystrophin. They did not have the protein. In fact, they were specifically selected to ensure that they didn't have the dystrophin protein, so that there wouldn't be a confounding factor that you could say, oh, they're really Becker's patients. They're not Duchenne patients. And then they studied this group, and they analyzed all 419 boys, looking for genetic factors that would explain how this worked. What geneticists always want to find, they want to find what they call a compensatory gene. So it's a gene that they didn't think of before that fixes the problem with the first gene, and then they can do gene therapy, because that's what geneticists do. It's like surgeons do surgery, geneticists do gene therapy. What they identified instead was that stem cell polarity, the process I described earlier in response to one of Martin's questions, was actually what was going on. That these children somehow had restored stem cell polarity. In other words, their stem cells were able to make progenitor cells. And they concluded their work by citing Dr. Rudnicki, my co-founding partner, extensively. And then they concluded, and I've pretty much quoted their conclusion, so I'm not putting words in their mouths, that affecting polarity, changing polarity, represents an important area for study, and two, that their beloved gene therapy approaches will not correct that problem. In other words, if somebody could have written an advertisement for us, this would be it. So this study validates that humans, if they can regenerate muscle, they can keep walking, despite having Duchenne muscular dystrophy. This is a remarkable, remarkable finding. And it validates our approach totally. Now, we still have to develop the drugs and so on. I'm not saying that we don't have to do the work. This really provides human proof of concept of our thesis and truly validates Dr. Rudnicki's findings, which is, it's thrilling for us, and it gives us massive confidence and more encouragement to keep moving ahead.
Frank, if it's not a genetic cause, did they come up with any indications as to why those boys had a difference in their polarity, diet, lifestyle, pollution, or who knows what? Or like,
so there are always sort of biological accidents. Okay, so in this particular case, in these children, the single factor that appears in our reading of the paper, the single factor that appears to be the greatest influence is a particular protein that's part of a group that affects polarity. And it's one that we also consider part of the work that we do. And they cited this particular polarity factor extensively and said basically what's happening in these boys is that they have some type of compensating mechanism that is allowing those polarity regulators to work in spite of not having dystrophin, which is the key to it all that Michael Rudnicki discovered. What we're expert at as Satellos is exactly understanding these regulatory pathways. And so I can't say too much because we're actually in the process of filing intellectual property around a lot of this. They've actually provided evidence for some of the things that we're going to be filing patents on. That essentially, if you can restore these regulators of polarity, that you can regenerate muscle. And that's what this shows, absolutely shows. It's just astonishing. It's astonishing.
fantastic. All right.
And it's really the first time we've seen that somebody's asked the question, why are these outliers able to do it? It's like a Malcolm Gladwell kind of question. Why do the outliers, how did they do it? And this is what they found. And it's, remarkable. And, Michael's been saying this for, since 2016. So takes a long time to be recognized sometimes. So we think it's just fascinating. And what it means is we think that this will really allow us, as time goes on, to position the company with, other players in the field, with other major patrons in the field, and so on. So I think this will really give us a lot of ammunition and ability to get noticed. And ultimately, when we're further along, I think we'll be a very attractive candidate for some of these larger players in the field who want to have this kind of technology in their company. So what are we doing as we move along? We're in the process of inventing and advancing drug candidates. So our goals for 2022, and I'll speed up a little bit, Martin, if I may, because I want to make sure you have time to get the questions from the field. The goals for this year are to get that compound nailed down and begin the process of what are called pre-IND studies to move forward in 2023 into human clinical trials, and along the way developing translational biomarkers so that when we're in the clinic we can get readouts very early on, whether we're on that mechanism that I described of shifting to progenitor cells. And we're in the process of identifying sort of the next areas of study in terms of disease after our first program.
All right, I just quickly, I am aware of time as well, but so just to clarify on the last slide, by Q3 or Q4, you hope to have one or maybe a couple of drugs identified as your best candidates, and then start that
Yes.
trial process?
Yes, well, it's called pre-IND. So it's a two-step process. You got to put the package of data together that the FDA approves. That's called an IND. So
Yeah.
you call it pre-IND, and then to begin the trial. So, and we look at this as kind of seamless. Once you start, then it's step by step. I mean, you can't screw it up along the way, but it's a step-by-step process.
And then hopefully you submit it, I guess, to FDA by the end of 22, and then sometime in like 3-6 months in 23 that you would get the IND from FDA.
Yes, it'll take a little longer than the end of 22 to get it submitted. They take a little longer to put together, but we'll have it submitted in 23, and the approval process is fairly quick if they approve it. And of course, you meet with them along the way. So you don't do this blind. You meet with them before you submit, and you make sure that they like what you're doing. If they have any questions, you can address it. If they have any advice, you can deal with it. They don't tell you yes or no up front. They just tell you, what won't work mostly, but they give you some guidance. And so, you don't expect a surprise as you move along. You expect it to become seamless. And, we've also aligned ourselves with a group called Parent Project Muscular Dystrophy. They invested in our company. They're the main advocacy group for this disease in the United States. They have extensive networks in Washington and with the FDA. They're involved, in fact, in developing the clinical trial protocols for this disease area. And so, we wanted to align with a group like that so they can help us. So as we go along, we won't be doing this all by ourselves, just a bunch of, schmoes up in Canada. We're going to have really serious help to move this along.
When you say FDA responds fairly quickly, that's within like 1 or 2 quarters, or like one or 2 months, or?
Yeah, it's a 30-day turnaround. So.
Oh, wow. Okay.
Yeah. So I mean, once you get into the process, it's a 30-day turnaround. It's actually the same with Health Canada. It's a 30-day turnaround.
And sorry, just quickly on the biomarkers, those are biomarkers or indicators you can tell quickly if it's working or not working, and it's like a quick gauge of, okay, if we see this, then we're on the right track. If we don't see it, then we're not on the right track.
Yeah, it's like that slide that I showed you. First you want to see you're on mechanism, so you're getting the progenitors. So we're looking for a marker of those progenitors. Well, we found a marker of those progenitors would be a better way to say it. So now it's making sure we can validate it. So getting the marker, and that marker, then says if you've got the marker, then you're going to get the muscle fiber. If you get the muscle fiber, you're going to get the functionality. So it gives you the way to say to people, this is predictive. If we're on mechanism, we can have confidence that that translates to effect. So mechanism and effect is what you want to show. And that's what's tied in with biomarkers. And that's why it's so important that if you can show mechanism, and mechanism can be plausibly argued to lead to effect, then you've got something. And what I just showed you from that study that the geneticist did, it absolutely demonstrates that if you produce progenitors, it has an effect. It absolutely demonstrates that. No matter what else it does, that it does absolutely.
So crudely, the biomarker would be how many progenitors there are, and if you can count the progenitors, you're basically on the right track.
Yeah, it's not exactly that, but it's essentially that. Yeah.
Okay.
Yeah.
Thank you.
Yeah. It's essentially that. So we've developed a proprietary platform to study from the muscle. We take tissue, we can look directly at that environment in which the stem cells reside. And this allows us to define these various biochemical pathways. We've named some of them here in the middle on the right-hand side from our platform called MyoReGenX. And we can look at how they influence either self-renewal or that differentiation process. So in time, we want to create a pipeline of disease-specific modulators of this stem cell process of polarity.
Your platform technology or knowledge base, where you can hopefully target other diseases and indications, that is essentially a stem cell process or mechanism that is the foundation of your knowledge technology?
Yeah. So I mean, the expertise we've built is around understanding the stem cells and
Okay.
understanding the regulators of those stem cells. So how do they get regulated? And everything in our body is regulated. So how do they get regulated? That's our expertise. And so we've designed a system that allows us to look directly at those stem cells and at the processes that regulate them. And that's what we focus on. And, as we build the company, beyond 2022 and into 2023 and afterwards, it starts with a phase 1 clinical trial in Duchenne and then quickly moves into more advanced clinical trials over the balance of and into 2023 and into 2024. We will look at additional disease indications. As I mentioned, there are other dystrophies, and then we will look at a second program down the road. So we've tried to just stage out over a 2-year period how this company could build out. And we believe that once we're in clinical trials in 2023, we will become of real interest to potential partners and potential acquirers, because now we're on to, proving that that what we have has the potential for an impact.
Frank, as your background as a VC, so on the money side of drug development and healthcare, what do you see as being sort of the key milestones or value or de-risking events or value creation events? Like when you get your IND valuation kind of goes up, what are the major steps over the next couple of years?
So it has a lot to do with the audience. So when we're talking to the really significant institutional type investors in the U.S., people like Bain Capital and these other really major players, they're looking for certain things, and what they're trying to do is gauge from wherever you are what you have and the likelihood you're going to get to the next phase and generate value. So the kinds of things that really matter, and at least in my opinion, moving forward here, number one, a drug candidate. That's absolutely the most important, is to nail down that drug candidate, which is why it's highlighted on a slide of its own. Once we have that, then having the ancillary information about the potential for biomarkers that give us a readout on efficacy, the idea that we understand multiple pathways and how they come together, and we understand the biology deeply so that we know that the mechanism can translate. All of those kinds of things are vitally important to potential institutions and potential partners as well. And that's why a study like the one that came out of Ohio State is so valuable to us, because it proves our case. And then the next thing is, knowing that the team is in place that can do all the work necessary for an IND and so on. So the investors those kinds of investors will believe, once we've got the package of information, that the next steps will flow. What they want to know mostly is we've got the team that can execute on it, and we understand all the potential hurdles along the way, and we have ways to deal with them. And so that's what that's really what we focus our time and energy on. And what I focus my time on as the CEO is making sure I'm talking to these people, and then making sure I'm putting in place the team that can deliver on it. Because I'm totally confident that this biology is not just new and unique, but I'm totally confident that it is profoundly important, and also that we will get to a drug candidate. I'm very confident that we'll get there. So now what I'm thinking ahead is a year or 2 years ahead, making sure that all the pieces are in place to deliver. So you move quickly from can you do something to will you execute? And that's really what these big boys want you to be able to answer. Is it fascinating and different and possibly world changing? I think we check those boxes. Then they want to go through and say, okay, so are you going to execute? And so that's what I'm working on. So I'm sorry if that was a long answer, but
No,
hopefully that helped.
that's good. And Chess, it seems like with your team that you have right now, at least on a pre-IND basis, they're super qualified. And I'm sorry, I forget your partner's name, but out of
Michael.
Ottawa, he seems very impressive fellow.
Yep.
Rudnicki.
Rudnicki, yeah.
How
Ukrainian
far,
guy. Yeah.
like, do you have the team to do the IND stage, or when, like, and because the
Yeah.
early stage scientists aren't necessarily the
Yeah.
trial scientists.
Absolutely right. So that's why, we started at the board level. We brought in people like Dr. Rima Alawar, who heads up exactly this type of a function at the Ontario Institute for Cancer Research and comes out of Lilly, and Bill McVicker, who is a pharma guy who specialized in that early stage development. And what we will be doing in the next 6 months is bringing on board a head of R&D who will be a development-oriented person. So we'll get that person on board early, and make sure that that individual aligns with the rest of the team and then can lead us forward from where we are. So that's the next big hire. And then after that, making sure you've got the right medical advice. But right now we have great advisors, Dr. Ronnie Cohen at Sick Kids in Toronto, who's a Duchenne physician, president of Sick Kids, Kevin Campbell in Iowa, and others. So we've got lots of good, strong advisors. And so now at this time, we use advisors to make sure that we're going in the right direction. And then when we're far enough along, then we onboard the right kind of people to lead.
So in 3 to 6 months, maybe we should be expecting a head of R&D
Yep.
that has like a very well-established resume.
Yes, absolutely. Absolutely. So I'm talking to people now, and we'll probably do a formal recruiting to make sure we cast a wide net. But yes. Mm-hmm.
Gotcha. Thank you.
Okay. And this is the team. It's we, from the beginning we tried to put really, really good people. Jeff McKay, I'll just call out Jeff McKay, the chair of our board of directors, who's based in Boston and has founded a number of companies, including AvroBio, where he's the founder and CEO. Originally came out of Novartis. Sridhar Narayan, who heads up our drug discovery efforts as VP and is a terrific medicinal chemist. We brought on a great board member in Adam Mustafa, who chairs our audit committee and is the CFO of a NASDAQ company called X4 Pharmaceuticals. I mentioned Rima and Bill McVicker, and Ronnie, and John McCall is a former very senior guy at Pfizer, chemistry guy. So, and Keish and Bill Jarosz, who joined us from the ICO team. So, it's a really, really strong group of people. And in fact, we had a board meeting this morning as a team, and we communicate well, and we get lots of great input. And I believe very strongly that you need lots of good input, and you need different voices and different backgrounds to make biotech work, probably to make any company work, but definitely to make biotech work. And that this is a little bit of history, but I think that's well known, and it's pretty clean cap structure. We have good advisors, Cooley's in the U.S. and Price Waterhouse on the accounting side, and BLG on the legal side, and there we are. We believe it's transformative science. It's super cool, and it matters. It's important, and it matters, and I think there's a great business opportunity here, and a great investment opportunity from a stock point of view, or a valuation point of view, I should say. I'm not selling stock. I'm just saying that from a valuation point of view, we think that this is a great market to be in, and we will be of interest to other players for sure.
Quick questions before we move on to the audience questions. Thanks everyone for being patient here. You've got, I believe it said $5 million of working capital. You're burning about $500,000 a month. That's about 10 months of runway you've got here. You were talking about NASDAQ in the second half of the year, or start the process of that. I guess is sort of the funding strategy of NASDAQ, and are there non-dilutive financing options that you have, just government grants or other things? Or I'm thinking that whole platform technology on stem cells, is there any way to maybe license that out to other companies to target different drugs to help fund your today's operations?
Yeah, so we're exploring all of those options, including our legacy assets, whether we can monetize some value from those, and we're looking at different vehicles to always extend our runway, and we have a number of warrants that are also on the board that may come into the money in the next little while, or longer during the year. And we can't rule out that we also, frankly, will need to top up our funds. But we're not in a situation where we need to worry about any dramatic action that we need to take. We can pace our growth. We can pace the financing needs in a way that's tolerable and allows us to get where we need to get to. And I'm very confident that we'll be building value quickly in the next year. That's the main, that's the key thing. So.
And I guess, do you expect your burn rate to go up substantially until maybe you start your phase 1 trials, or is it kind of this a good pace we're at for a while?
Yeah, we're at a good pace for a while. We can titrate it down or up as we need to, if we need to. I'm not anticipating that there'll be any dramatic change in the burn rate for the next little while. Towards the end of the year, that's when we plan to step up the burn rate. But that is contingent on also making progress with all of our milestones. So I think we're in pretty good shape to manage our situation.
Okay, great. I'm going to now switch to the audience questions. Thank you very much for everyone. We've got 13 questions here. I'll ask the questions. I think you've sort of addressed some of them already, but just pass it to you to make sure if you want to hit anything else on it. Can you update on biomarkers and drug candidates for muscle regeneration?
Yeah, so just to add to some of the things that I've said before, we have identified biological biomarkers of the stem cell activity that we're looking for. And we use them now in some of our internal laboratory work. And as far as the drug discovery side, I would say we're on target with the guidance that we've given previously, that we want to have candidates in Q2 that we're working on in animal studies, and we want to be in a position by, as I mentioned in the slide, by the fourth quarter that we're taking candidates forward to start the pre-IND work. So I think we're on target for that.
Right. Another question, Dr. Rima Al-Alwar, I hope I said that right, is surely on board to help refine and identify lead drug candidates. How close is the team to achieving this milestone?
So Dr. Alawar is a member of our board, and I've known Dr. Alawar for probably 15 years. So she does give us advice as well, and helps with making sure we're strategically doing the right things. But she's not a part of management of the company. I just want to clarify that in case somehow we created that misimpression. We are very close. We are constantly testing drugs. We've made hundreds of different variations of drugs to fit the purpose that we're trying to achieve, and we test them in a regular cycle. And we're doing that now. In fact, we'll get data on Thursday or Friday of this week from Shradar on the latest batch of drug candidates. So I can say that just to get back to what I said a moment ago, we're on target that this year we'll have drug candidates in the first half, and we will have, we want to be moving into IND enabling studies in the second half. That's the goal.
There's a question here regarding the share price. Can you talk about any sort of promotional or investor relations activities to add some volume and some good direction to the share price?
Yes. Well, I'm open to any ideas as well. Today's one of those activities, I suppose. Right now we're in the worst. This is the worst biotech financing environment I've seen since the year 2000, and I lived through the crash of NASDAQ in 2000 and got a lot of scar tissue from that when it happened, when NASDAQ dropped over 80%, almost overnight, and took years to recover. So there's a lot of things going on here that frankly we just can't control, and we're getting buffeted by a lot of stuff. But to the point of building demand, I'm regularly out speaking to people. I'm regularly communicating and trying to communicate with major players in the field, major investors that have invested in our company, working on different approaches and strategies to try and expand the network and so on. And it will take a bit of time, but we are working on it. And right now, I think it is a little bit of a valley that is not all of our own doing. We are trying very hard to make sure we're positioned that people understand what we're doing is different. What we're doing is profoundly important and can have a huge impact, and that we're getting very close to having meaningful progress that will translate to value. So, I'm hoping people will pick up on those messages and start accumulating some stock.
And just to clarify, if like the biotech market, along with so much of the spec and high tech market, really peaked last February, March, and so forth.
Yeah.
It was super frothy. And
Yeah.
since then, it's been a massive correction, and lots of people are taking it hard right now. You're in good company, so to speak.
Absolutely. So all the major biotech indexes are down 40 to 60%, depending on which ones you look at. They've all been really crushed. Really substantial companies in the U.S. have been hammered, just absolutely hammered. And of course, the Canadian biotech market is thin compared to the U.S. So the knock-on effect is even more serious. But more fundamentally is that in this environment, we've got all of these other factors. You've got the situation in the Ukraine. You've got inflation. You've got all sorts of other issues that caused by various views of action or inaction. And you even see it in the big board, and you see it in the Dow. It's up and down on a daily basis. So we need a little bit of stability, but that's not stopping us from being out talking to people and pushing as hard as we possibly can. And I'm going to keep at it. I'm just going to keep doing it, pushing and pushing and pushing. And I believe in this story. I've worked on this project with Michael now since we first started talking about this company when he made that publication in 2016, and we formed the company in 2018. So what's that? 6 years I've been involved in this project to make this happen, and I'm not going to stop until we succeed. So we're on it.
There are a couple of questions here on ICO, the assets and time frame and monetization. Is there anything you can add to that?
Well, I can say that we are exploring ways to try to monetize these assets. I would also point out, to be fair to everybody, that when we acquired ICO in August of last year, those assets, the 008 program and the 019 program, had really not had any activity in over 2 years. Those are assets that have been fallow for essentially 2 years or longer in some cases. And so there's some rehabilitation we have to do, and there's some try and repackaging, repositioning, or opening up new dialogues, and we're working on it. So if we can monetize, we will, and when we can do something, we will let the market know as soon as possible.
All right. You have an MOU with SkyMount that is 14 months old. Any update there?
Yeah. So in fact, I spoke with the CEO of SkyMount yesterday. So, we are trying to put together something with them that would see them, taking on some rights to move the assets forward, to move the AMP-B program forward in particular areas. I can't really say much more than that. At this stage, I would say these, we're at a point where I can't say where it's a slam dunk we'll do something. There has to be value on both sides, and Satellos has to get enough sufficient value as well. But I'm encouraged that we'll be able to do something with SkyMount.
Right. There are a couple of questions, and we've addressed it, but I don't know anything else to add to it in terms of joint development agreement or partnerships, or anything more you can add on that front?
Well, we're always talking to people about the company, and I do have a lot of experience over the years with partnering and with various types of transactions, and also the timing for those. We are probably now, over the next 12 months, in the highest value build we could ever be in, which is getting from an idea to the point that we're actually going to be putting a drug into human beings. And it's during that period that we want to create as much value as we can and start to get people interested about what we're doing. but not show them we're too hungry, if you know what I mean. So we're always talking to people, and my objective is to have multiple options for crystallizing shareholder value over the next 12 to 24 month period.
All right, and there is a question specifically on NW Pharmatech.
NW Pharmatech. Okay, I'm stumped.
Okay, so am I. Then I will.
I'm sorry, I'm stumped. I don't know what that is specifically referring to.
All right.
Is it NatureWorks that they're referring to? Is that the CBD project that they're referring to?
I don't know.
I'm sorry. Okay.
All right. Frank, we are almost an hour and 15 minutes into this, so I think that was thorough. I feel like I know what's going on in the pathway. It was, and sorry, there's a comment. Yeah, it was, I guess, regarding to the CBD.
So we'll be giving an update in the next within the next couple of weeks on NW Pharmatech.
Okay. All right. So yeah, to reiterate, I think that there was a big transfer of knowledge here. I hope the audience enjoyed it. I feel like I've learned a lot and understand the pathway of what's going forward. And so Frank, thank you very much. And any final comments to wrap things up here?
Well, Martin, thank you so much for this opportunity. I really appreciate the chance to speak to such a large group of investors. I appreciate the questions. Thank you for the questions. And I hope you did get a good sense of what we're doing, and at least our excitement with what we're doing, and our view of the importance of what we're doing. So thank you so much.
All right. Thank you very much, Frank.
Okay.
Have a great day, and hopefully we'll talk again in a little while as some of those milestones get knocked down.
Absolutely. All right. Thanks a lot. Take care. Bye now.